Hello and welcome everyone to this plenary session at Future of Back Pain Therapy. It is my pleasure. My name is Alex Breen. I am Senior Research Fellow from the AECC College in Bournemouth, and I'm joined by Valerie Sparks, Professor Valerie Sparks, Director of the Biomechanics and Bioengineering Research Centre at Cardiff University. It is our privilege to be moderating these sessions. Valerie's going to give us an introduction to Christine shortly, but I just want to say, if you have any comments or questions, please drop them into the chat box, and we'll send them throughout the session.
Okay.
Over to you, Valerie.
Welcome, everybody. Thank you for joining after lunch break. I'd like to introduce you to this plenary session, The Future of Back Pain Therapy, by Professor Christine Lemaitre. Christine is an internationally renowned researcher investigating musculoskeletal disorders and novel therapies, specifically in relation to disc degeneration. She completed her BSc Honours in Pharmacology with industrial experience from Manchester University in 2000 and her PhD from Manchester in 2003. And she currently works at Sheffield Hallam University. Her research focuses on the understanding the pathogenesis of intervertebral disc degeneration with the aim to develop new therapeutic approaches to target abnormal cell biology, which results from intervertebral disc degeneration. We look forward to hearing about this exciting new approach to back pain. Thank you.
I'd like to thank the organisers of BritSpine for the invitation to talk on the future of back pain therapy. I'm Professor Christine Lemaitre, and I'm based at Sheffield Hallam University, where I lead the Tissue Engineering and Biomechanics Research Group. The hydrogels which I'm going to talk about today, I'm an inventor on, and therefore could trade as a conflict of interest. So first of all, if we want to develop new therapies for back pain, we need to understand where the source of pain is, because of course that is the symptom that patients come to. They're in pain. They want us to target that pain and prevent it. But back pain is a very complex condition. It's multifactorial, and whilst there may be biophysical underlying causes, these are interacted by the genetic makeup, environmental factors, social factors and psychological, and also further complicated by comorbidities. All of these things together generate the symptom of chronic low back pain. And this means that actually for the majority of low back pain patients, they do not have a well understood pathological cause. And this is a current major hampering of developing new treatments for back pain. And we need to get much better at patient stratification and working out what exactly is the underlying cause of the patient's problem, so then we can then target and treat the individual with potential new therapies. So where could the source of pain be coming from? There was a very interesting study 30 years ago now, where they basically looked at the response to different tissue stimulations during operations and looked at which tissues, when they poked them, basically simulated the pain that represented the standard pain of that patient. So obviously they could only probe tissues which were available to them, so they couldn't do the nucleus. But all the other tissues that they actually probed, you can see those with the asterisks, stimulated the pain which was similar to that pain that the patient experiences in certain individuals. But importantly, different patients experience pain probed by different regions. And this actually is the major problem, in that there is a multitude of potential sources of pain which will vary from patient to patient, and they could be the muscles or ligaments, they could be the intervertebral discs or facet joints, and therefore we need to improve the diagnosis of individuals and the stratification and full investigation of what could be the cause, so we can then target the therapies. Now, the further issue is actually at the moment there isn't cures for many of the causes of back pain, and therefore the thorough investigation for diagnosis is not warranted, because there isn't actually a therapy available if you identified that as a cause. And that, I think, is one of the major limitations, because we're not actually investigating what causes pain in a number of patients and it's been put down to non-specific low back pain. That's not good enough. So I call this as a call to arms to those of you that work in diagnosis, biomarkers, improved imaging. We need to do better at actually developing better understanding of what causes the pain, so then we can go on to develop new therapies. Now, one potential source of the pain for low back pain is the intervertebral disc. And that is the area that I research, looking at what happens with the pathogenesis of disc degeneration and how we can target these pathogenesis of disc degeneration to the development of new therapies and treatments. So the discs degenerate, and they can occur during normal aging process, but also can get accelerated disc degeneration at early age. And this is a classical feature here. You can see that this disc here has prolapsed, causing a large herniation of material pressing on the local nerve roots. And this individual was actually me. And I can assure you this was painful. I have had discectomy operation to remove this herniated material, which has improved matters, but still I have back pain because my disc has not been treated. All that's been treated is the material that is extruded, so it's no longer pressing on the nerve root and causing sciatic pain. But the key issue with disc degeneration is the fact that disc degeneration is also visible on imaging of asymptomatic individuals. And therefore, targeting and identifying the intervertebral disc as a source of pain, when you also see the same features on imaging of asymptomatic individuals, is problematic. And actually, there are some really fascinating studies on new imaging modalities which could have the potential to show differences between symptomatic and asymptomatic individuals. So there is a lot of research still needed, but there are certain things which have been identified to be different between asymptomatic and symptomatic individuals. And in fact, if you look in younger individuals, so those under 50 years of age, then actually the association of MRI findings in disc degeneration and low back pain is much tighter. And this is probably because actually, if you look at the under age of 50, the features you're seeing are those of an accelerated disc degeneration, not the normal process of aging. And it's important to dissect the differences seen with accelerated disc degeneration and that of a normal aging disc. So how can disc degeneration actually lead to low back pain? Well, the actual degenerate disc itself causes decreased loss of height, which will alter the biomechanics of the surrounding tissues. This can cause nerve root compression. It can cause the release of catabolic factors and nerve factors, which can cause the stimulation of local nerves and pain. And of course, it alters the mechanics of all of the tissues around, stimulating increased compression or rubbing, etc. And there are certain features, such as modic changes, which can be seen in these vertebrae here, which are the loss of bone within adjacent vertebrae. And these modic changes have got a good association with linking disc degeneration to low back pain. The actual pathophysiology of modic change is not fully understood, and it needs a lot more investigation to work out what causes these modic changes. Are the modic changes infection? Are they generated by the catabolic factors produced by a degenerate disc that then affect the bone adjacent? Or is it a bone defect that then has the knock-on effect on the disc? And actually, this is probably further complicated by, it's probably all of those things, but in different patients. And this is why we need better understanding and stratification of patients so we can target therapies. So what about the disc itself? Is that actually a source of pain? Is it innervated? And indeed it is. So whilst a normal disc is known to only have nerves around the very outer layer of the intervertebral disc within the annulus fibrosus and within the cartilaginous end plate, during disc degeneration, those nerves ingrow. So it's shown back in 1997 by Professor Fremont at Manchester University that nerves could be seen right into the nucleus pulposus of intervertebral discs from patients undergoing chronic back pain. And this has been followed up by multiple studies which have shown vascularization within the disc or within surrounding tissues, and that the disc could cause an increase in innervation during degeneration. We demonstrated that this could be driven via the production of nerve growth factor, which is stimulated and produced in the blood vessels in the outer annulus fibrosus. So here we have the outer region of the disc and a blood vessel, which is expressing lots and lots of nerve growth factor. All the little black blobs is where you've got NGF being produced. And these blood vessels associate with nerves coming in. In addition, these blood vessels and associated nerves are often associated with tears within the tissue. And indeed, if you look at discs, there is a lot more nerves than you previously thought. So we did a previous study where we actually looked at over 200 disc samples and found there was these fine nerves right in the nucleus pulposus regions of the intervertebral disc. And actually, we saw nerves in deeper regions than the blood vessels. So it actually appears that nerves are able to transmit and migrate into the disc further than blood vessels. But they always seem to do it in association to disrupted tissue or fissures. And this is not really surprising because it's known that the proteoglycans that are produced within the disc are also inhibitory to nerve fibers. So therefore, in these tissue fissures and cracks where you've got a loss in the extracellular matrix, this then doesn't inhibit the nerve ingrowth anymore, and it actually enables nerves to come in. But nerves don't just move somewhere just for the sake of it. So what's actually calling the nerves into the central regions of the disc? Are there factors that the disc cells themselves are producing that stimulates the nerves to grow in, but also could there be factors that stimulate the nerves to sense pain in a heightened way and therefore link to increased discogenic pain? And indeed, there are a number of studies which have shown that the cells within the disc synthesize nerve growth factor, which is a factor that can pull the nerves into the disc, and also factors which can sensitize local cells to pain. So brain-derived neurotrophic factor and other neurotrophins are expressed by disc cells, and these are factors that, if stimulated by local nerves, will cause an increased pain sensation. We've shown that these are produced in high numbers of discs from degenerate patients, but also in non-degenerate disc tissue, so those graded 0 to 3, but these are ones which are taken from painful individuals undergoing herniating material. We see increases in NGF, brain-derived neurotrophic factor, VEGF, which is a vascular endothelial growth factor, and interestingly, substance P, which is a factor which will increase the sensitization of local nerves. These factors are stimulated by treatment with the cytokine interleukin-1. And interleukin-1 appears to be a pleiotropic cytokine within the disc, causing increases in NGF, substance P, both at gene and protein level, and therefore this could be a key factor in regulating for treatment of disc degeneration. So could one of the reasons why you have patients with no back pain, well, individuals with no back pain, but they have degenerate discs, could substance P be the differentiating factor between the painful and the non-painful discs? Meaning that once you've got substance P there, apologies, degenerate discs, and therefore substance P could be a potential biomarker that we could investigate to see whether it's produced within the CSF, for example. And the nerves within the degenerate discs actually produce high levels of substance P, demonstrating that the nerves within a degenerate disc from an individual with pain is heightened to sensing pain. So what is sort of disc degeneration more generally? So if we've shown that we do have nerves which can grow during the process of disc degeneration, I've mentioned that that often leads through the fissures within the disc because of the decrease in extracellular matrix components in the disc, and therefore enabling those nerves to grow in. So during disc degeneration, you have a progressive loss of the normal matrix within the disc, and the disc becomes more fibrotic and less hydrated. This is principally due to the decreased levels of proteoglycans, which are negatively charged and hold lots of water. So in a hydrated young normal disc, you can clearly see the differentiated region of the nucleus pulposus, both on this image and also the macroscopic image from cadaveric discs. And this gelatinous region of the nucleus pulposus contains high amounts of proteoglycans, so holds onto lots of water. This is surrounded by the concentric layers of the annulus fibrosus, which is high in collagen type 2 and a highly ordered structure to maintain the nucleus pulposus where it should be and retains the hydrostatic pressure of the nucleus pulposus. During degeneration, the proteoglycan levels decrease and the MP region becomes more rich in collagens, which you don't really want. It's not going to be a hydrated region, and you can see that it becomes much more stiff and you get these cracks and fissures forming within the degenerate disc. So if we look at the disc in a little bit more detail, the nucleus pulposus, this central region here, is composed of rounded cells which produce the collagens and the proteoglycans, but they also produce factors which break down these collagens and proteoglycans, known as matrix metalloproteinases and ADAMTSs. And these are enzymes which are important during normal homeostasis, but this is kept in check by regulation by growth factors, which will promote the synthesis of the matrix, and also catabolic factors such as interleukin-1 or other cytokines, which produces the cells to produce matrix degrading enzymes, which enables the remodeling of the disc. So this is in a normal disc. You have highly hydrated proteoglycan structures bound together in a collagen meshwork, and it's all maintained by nice, healthy MP cells. Within the annulus fibrosus, you have more elongated cells and the more collagen type 2 in the central regions of the annulus fibrosus, change into collagen type 1 in the outer region, small amounts of proteoglycans, and also elastin, particularly in the interlamellar cross bridges. In the normal disc, you have nerves and blood vessels only in the very outer layers of the annulus fibrosus. But even a healthy disc is actually quite a harsh environment for these cells to survive in. It has low levels of oxygen because of the low amounts of blood supply and also low nutrient supply. There is a high osmotic concentration, which means there's high amounts of salts and water within there, and it has a slightly acidic pH. And of course, the disc is a loaded structure, so the cells are used to being under dynamic load, which means they're constantly compressed and under tensile stresses in the outer annulus. During degeneration, we see that you get lower oxygen concentration, decreased osmolarity, a further decrease in pH, and low glucose, and also altered dynamic mechanical load. Well, if we look at the cells, you get cellular clustering or cell senescence, and the cells take on a much more catabolic phenotype, producing heightened amounts of cytokines, particularly interleukin-1, IL-8, and IL-6, and TNF. And these cytokines lead to the production of matrix degrading enzymes and also neurotrophic and angiogenic factors, which call nerves and blood vessels into the disc, which can even be seen in the central region of the nucleus pulposus in the presence of fissures. In the annulus fibrosus, you also see a lot of these factors calling in nerves and blood vessels, breaking down the extracellular matrix and causing fissure formation within the intervertebral disc. But what is a key driver for these processes? So we've shown previously that a key factor that seems to be involved during intervertebral disc degeneration is interleukin-1. So interleukin-1 is produced by the native cells within the disc. The native cells of the disc also sense and respond to interleukin-1, expressing the receptor. And these cells respond via producing neurotrophic factors and vascular endothelial factors, calling nerves and blood vessels in. They cause a decreased production of proteoglycans and collagens, leading to decreased matrix synthesis. They increase the production of degrading enzymes and also other cytokines and chemokines, and they also have been shown to increase cellular senescence. But one argument is always, is this cause or is it effect? So utilizing an animal model where we knock out the natural inhibitor of interleukin-1 receptor antagonist, we see that when you remove the control of IL-1, you get spontaneous intervertebral disc degeneration within these mice. This mimics the effects that you see within human discs, with increased levels of interleukin-1 and increased levels of matrix degrading enzymes, and also an increase in senescence, i.e. you have a decreased proliferation capacity within the knockout mice compared to wild type mice. But what's the trigger for these changes in cellular homeostasis? This still isn't fully understood. Genetics certainly has a place, and what that genetic risk factors do is under multiple studies. Could it be an abnormal loading, for example, a fall or trauma during early in life? It's well known that trauma can lead to an increased incidence of osteoarthritis. It isn't fully known for intervertebral disc degeneration. And also there's lots of environmental factors such as lifestyles or potentially infection as well, which could lead to triggering of this catabolic phenotype that's seen during disc degeneration. One of the key things for intervertebral disc degeneration is that it is a vicious circle. So whichever point you come into, it then has a knock-on effect of the next stage. So for example, if you have a biomechanical alteration, trauma, for example, this can cause a different effect on the cells. The cells will then produce different levels of extracellular matrix, which then means they aren't able to respond as well to load. And therefore you have this constant traumatic event going around. And of course, the response of cells can change regarding to the genetic makeup of those cells. So it's not a simple situation, and we do need to do more work to understand the pathogenesis of disc degeneration to try and develop new therapies. So what are the kind of novel therapies that are being looked at the moment for low back pain, which results from disc degeneration? So there is still a need to gain a better understanding of the normal disc and its pathogenesis. But the key research areas that people are looking at, and in fact, a lot of these are now progressing to clinical trials, targeting the disc and replacing dysfunctional cells with the prospect to restore mechanical properties. And of course, we must inhibit those processes of disc degeneration. Otherwise, any cells you put in there are just going to be more fodder, basically, to produce more extracellular matrix degrading enzymes, more catabolic factors, etc., if you don't create the correct niche for them to restore. So in order to target the intervertebral disc, we're likely to need a combination of things to restore the mechanics, inhibit the catabolic environment of those neurotrophic factors that are stimulating pain, and this could be done via stem cells, biomaterials, or even gene therapy. And it's likely to need to be targeted depending on the stage of degeneration. So at mild degeneration, it may well be sufficient to simply inhibit catabolic factors or add growth factors or a nucleus pulposus regenerative cell source. Or you might want to combine that with the biomaterial system. In mid-degeneration, you may want to combine those different features together to provide an improved restoration strategy. You may also need to combine it with annulus fibrosis repair as well. And then the end stage degeneration, you may need to either replace the disc altogether or induce fusion, but we could probably do more smartly, so with single injections to induce fusion within the spine itself. And it's really exciting to see there are a number of clinical trials which are either ongoing or completed, looking at targeting the biological processes of disc degeneration or restoration. So there's been clinical trials in anti-TNF, which is an anti-catabolic factor, GDF5, which is the growth factor system. There's various clinical trials ongoing for stem cell therapies around the world currently, and also the use of cadaveric discs to isolate human intervertebral disc cells, which can then be grown up in culture and put back in to patient discs. And also there are inhibitors which are being looked at, which again will inhibit the catabolic processes of disc degeneration. So what we've been looking at is a biomaterial system, which is liquid above body temperature, and it has a very low viscosity, similar to milk. This means we can inject it through a very fine needle, and you'll be able to see this coming through with some red dye so you can visualize it. And as it cools down to 37, which can be seen in the glove box here, it instantly gels. So this could potentially be done as an injectable therapy as a day case, which would reduce cost massively. You can see it's gelled instantaneously and even filled the needle track where we injected it. This hydrogel system, we believe, has a three-pronged attack. It has the potential to restore mechanical properties. The hydrogel has a similar mechanical properties to native healthy MP tissue. And this is a system under compression here, where if you measure the MP gel, which is the name of our hydrogel, it measures an elastic modulus of 3 to 5, whereas human nucleus pulposus tissue is 3 to 8 kilopascals, very similar. And when you take a bovine disc and just stab it with a needle, you don't see an effect. If you induce degeneration via the degradation of the matrix using enzymes, you see a decrease in the mechanical properties of the disc. And then when we inject that degenerate disc with the hydrogel, you get an immediate restoration of the mechanical properties. So this is really exciting. We can also inhibit process of disease. So I mentioned that interleukin-1 was a pleiophoric cytokine produced by disc cells. Our hydrogel system can inhibit the production of interleukin-1 and also decrease the sensitivity of cells to IL-1 by downregulating the receptor. We can also use it to deliver a regenerative cell source. And we've previously shown that if you culture stem cells within our hydrogel system, we can tune them to disc cells. The hydrogel can be easily injected into intervertebral disc tissue. This is a cadaveric human intervertebral disc, which we're injected in with a hydrogel. We've incorporated iohexol so you can visualize it during the injection, and hopefully you'll see the black substance here, which is our hydrogel filling all of those natural cracks and fissures within the intervertebral disc. This can also be visualized on MRI. So pre-injection, you can almost make out the kind of cracks and fissures within the disc. And then post-injection, you can see the hydrogel shown here in white due to the high water content, that it's filled these little cracks and fissures. Excitingly, when you inject human cadaveric discs with the hydrogel, you get an immediate increase in disc height and also an immediate increase in the storage modulus or the mechanical properties of those discs. Now, the last thing you want to happen is when you undergo load is you don't want the hydrogel to come back out again. So we took those same discs and then performed failure testing, which is basically where you apply a compressive force, and these videos are showing the discs undergoing the compressive force until the disc either herniates, which you'll see in a minute here. It's about to go in a second. There it is. So there you see the nucleus pulposus tissue herniating, or the disc just fails itself. And you'll see on that one there just how much it had actually compressed before it failed. Now, what was really exciting is whilst we made every disc to fail, the level of failure is actually higher than that generally seen for discs of this age. And in not one of the discs that we tested did we see any extrusion of the NP gel system. We did see a forced herniation in one of the discs, which is probably due to a fissure that was within this naturally degenerate disc tissue, but that herniation did not contain any of the NP gel. So what we've also done is taken this into a model of degeneration. So this model is using goat discs cultured under daily loading regimes, where we can then mimic the normal loading pattern that discs see within the body. This load is applied for a period of a week. And what we've shown is what we can do is we can take discs and preload them. So we load them for 3 days. Then we can then inject them with an enzyme which breaks down some of the matrix, but not all of it, and then apply a 7-day daily loading regime. And what we showed was when you apply this load once you've destroyed some of the matrix, we see a phenotype very similar to human disc degeneration. So we see a decrease in the mechanical properties of the disc, we see fissures and cracks forming, and we see the cellular behavior of the discs changes. So within these enzyme-treated discs, there is a decrease in matrix production, so decreased aggrecan, collagen type 2, and an increase in the anabolic factors, interleukin-1, interleukin-8, anti-S4, MMP3, which are the degradation enzymes. Also, we saw an increase in NGF, which pulls in nerves, and an increase in VEGF that pulls in blood vessels. So using this model system, we've then taken that induced degeneration and then injected in our hydrogel. So here you can see that after 3 weeks of culture within that loaded system, the degenerate disc is almost obliterated. We probably went a bit too far. So you can see large cracks and fissures within the disc. The cells within it are starting to change their behavior. And actually, in a number of the discs, it had dissolved away quite a lot of the nucleus pulposus. Conversely, when we injected NP gel, either alone or in combination with stem cells, we see excellent restoration and repair. So the region here in the dotted lines is the nucleus pulposus gel, and the surrounding tissue is the actual native NP tissue. And excitingly, what we saw is within these regions here, which were the gel region, the native disc cells migrated in, and they then produced extracellular matrix within the nucleus pulposus gel, regenerating and restoring this tissue. We saw significant increases in the levels of aggrecan and collagen type 2 following the injection of the NP gel systems, either with or without stem cells. And we also saw effects on the catabolic factors with downregulation of matrix degrading enzymes and also downregulation of catabolic cytokines. So most recently, what we've been doing in collaboration with Lisbeth Haugland's group in Montreal is taking human cadaveric discs and growing them in a similar system, which can maintain discs under physiological load for 3 weeks. So here we take the discs, size them out, and undergo a washing procedure that washes all the blood and debris out of the vertebral end plates so that you can have good diffusion of nutrients through the end plates, much like you would within the body. This is then undergone loading within a loaded disc culture system and maintained for 3 to 4 weeks. After 3 weeks, we just double check the viability, and as you can see, the loaded discs have improved viability from unloaded discs, and the level of viability within these discs is excellent and shows that it can be a good system for maintaining human discs for long periods of time in culture. So using these discs, we've then taken naturally degenerate human discs and either left them alone within the culture system. We injected 500 microliters of NP gel or 500 microliters of NP gel together with mesenchymal stem cells. Immediately after injection, we imaged them using mid-axial T1 row MRI imaging. And as you can see sort of cracks and fissures within these discs, slightly more yellow regions within those injected with the hydrogel, but don't look too different to the control. However, once you've cultured them for 4 weeks in our loaded disc culture system, you can see an increase of intensity on the MRI imaging, showing we've got an increased retention of water, most likely due to an increase in proteoglycan content. We're currently undergoing the histological examination of these discs and hope to have some data on that soon. During the loading parameters, we took inline disc height analysis to look at the biomechanical responses of the discs. Looking at the MRI imaging, you can see really clearly that in some discs we had large cracks and fissures within the discs, that following injection with the hydrogel, you can see it scantily filled it. Within these studies, we limited the volume of injected hydrogel to 500 microliters, which, as you can see, does not fully fill the voids within the disc. However, following 4 weeks, this void has disappeared. You can now see that following 4 weeks, there's no void present, and actually the disc appears to have healed. If we look at the MRI quantification, we see an increase in the MRI intensity following the injection of MP gel compared to control discs. We're currently increasing the N number so that we can do the statistical analysis on this. Looking at the disc height changes, which gives you an indication of the mechanical properties of the disc, the discs undergo 2 hours of load, followed by 6 hours of static loading, followed by 2 hours of dynamic, and then 14 hours of static, representing a long night's sleep, I wish. So this is kind of quite a sedentary lifestyle, a bit like lockdown lifestyle, I'd probably say, where we only get once a day to exercise and then once a day cooking tea, etc. But what we see is that during the loaded periods, you see a decrease in disc height, which is to be expected. And actually you see a slightly more increase in more loss of disc height within those discs which contain the MP gel. But this is restored to a greater level during the static loading, demonstrating that these discs appear to have regained their compression and reswell capabilities that you should have within a healthy intervertebral disc. So this is all extremely promising and quite exciting. We're currently doing a histology and immunochemistry analysis on these discs and hope to have those results soon. But really, we need to transfer this now into a large animal model of disc degeneration as a prerequisite for clinical trials. Another option is the use of induced pluripotent stem cells. And we're working within a consortia project called BritSpine to look at the potential of induced pluripotent stem cells.
Ready to become cells of any type. We call these induced pluripotent stem cells. These iPS cells can now be retrained as juvenile disc cells, injected along with smart biomaterials, allowing them to thrive and regenerate the disc. Over the next 5 years, extensive laboratory and preclinical research will lead to the first clinical trial in veterinary patients. And maybe, just maybe, thanks to this guy, a cure is finally in sight for all of us.
So hopefully that shows just a little hint of some of the work that we're now doing, looking at alternative cell sources for the treatment of disc degeneration. So I'd just like to finish by acknowledging all of the various people that have worked with me on this project. So the postdocs and PhD students, collaborators and funders which have worked on this 20 years of research really into disc degeneration. I'd particularly like to thank all the surgeons who provide the clinical aspects and clinical samples and lots of conversations about what would be clinically acceptable for these therapies. And finally, just like to thank the funding sources, which without this work would not be possible. So thank you very much for the opportunity to talk, and I'd be very happy to take any questions within this discussion period, or do email me if you're interested after the conference. Thank you very much.
Thank you, Christine, for that fascinating talk. There is a lot of chat in the chat box. If anyone else has any questions, there's still time to ask them, so pop them in there and I'll go back and moderate through. Were there any ones that you would want to pick out first, or shall I read out some for you?
I'm happy to go through kind of as they've gone through. Yeah, so you pick what you want me to answer, Alex.
Well, okay, let's start at the top. First off, for my co-moderator, just a quick, thank you on your talk. You say quite early on in the talk that about a non-specific back pain, low back pain, is not good enough. What kind of term would you suggest to be a better one?
So I don't think it's so much as the term that's the problem. The problem is that we're not actually diagnosing patients in terms of what is causing their issues and what's causing their problems, and effectively just sending them away with the very generic diagnosis of non-specific back pain, which doesn't really help them, find therapies, actually know how to manage their pain and manage the back pain. So I think that's the key issue, and I see why it's done at the moment, because actually there isn't the therapies for many individuals and many patients. So there isn't the reason, I guess, to spend all the money on diagnosing them, working out what's the problem, if you haven't got a therapy for them. But we do need to do more work on that, so that as therapies do come through, we have got much better sort of patient stratification, so that then we can target patients for the therapies that are going to be useful to them.
So this wonderfully follows on to the next question that was in the chat, where Johan Hottel asks, sorry if I'm saying your name wrong, does it matter? I mean, for twisted ankle or headache, we don't really give these kind of specific causes of pain. So why should we worry about it when it comes to the spine?
Sorry, okay. I dropped out then. I wasn't sulking, I promise. So I think it matters when it goes from acute pain to chronic pain. So yes, with acute pain, it probably doesn't matter, because you can treat the symptoms and the patient will resolve themselves. In the case of headaches and twisted ankles, the majority of those individuals, it doesn't matter, because they can treat themselves. Can you hear me okay?
Yep.
Yep, super. But when it actually translates to chronic pain, it's not good enough just to give them painkillers or opioids for the rest of their life. We actually need to give patients a proper therapy that actually stops the pathogenesis, and that's why we need to know where the pain is coming from. That's what patients tell us they want to target. That's what they want to stop. We're only going to cure it if we know what causes it. It's no good just icing the cake if the cake's horrible, kind of thing.
So this is great because it's promoting lots more chats in the moderation section. Can I ask you can you go on to the next question? I need to go and do the moderation bit.
Certainly. So the next question I can see about the viability. So if it's from Val, how much does the viability function of the end plate play a role in disc degeneration? And there's this worthy investigation in combination of investigation of changes in the disc. So yeah, absolutely. The end plate is really important. And I think understanding what status that end plate is in is actually really important in also identifying what therapies may well work in individuals. So for example, in a patient where they have total calcification of the end plate, it's absolutely no point putting in new cells into that disc. They're not going to survive. The nutrient supply in there will be massively reduced. But there's also patients where you might actually have increased nutrient supply due to fissures or things in the end plate. So it's really, you need to look at not just the MP, the AF, you need to look at all of the tissues around to actually work out the stratification of patients to then identify what therapies are going to be useful. So the next question was around sort of the substance P. So can we reduce discogenic pain with anti-substance P? So I think that would be really good to look at. Substance P, and also I noticed there's a question on glutamate. So glutamate is also a neurotransmitter that increases pain sensitization, which has been found in discs as well. We personally haven't looked at glutamate, but I know. So I think therapies looking at those potential targets could be really interesting. But again, it's not looking at that initiating factor. So the substance P is increased because of something else that's going on in the disc. So if we only treat the substance P, then there's other neurotransmitters that will come in. And I think that's actually why, if you think back to the clinical trials on anti-TNF, it's why many of them failed, because actually it's not just TNF that's important. There's lots of molecules in there that are all important. So you need to kind of do a global inhibition, if you like, of those various factors to be able to get those successful therapies. Tell me when you want me to stop, Alex, otherwise I'll just keep running through.
So actually, there's some comments back on one of your earlier questions that have come through right towards the end of, well, very recently in here. Just going back to that non-specific low back pain. So persistent pain is multifactorial. This is bringing it back to a unidimensional cause of pain, which is what we're trying to get away from. Would you agree with that, or? And dropped again. I'm back.
I can still hear you when I drop. But yeah, you can't hear me. I'm certainly not suggesting that it's a unidimensional cause of pain, but what I'm saying is that we need to identify the physical sources of pain so that we actually understand what we can treat. You also need to obviously treat the psychological aspects. We can't really treat the genetic aspects that we need to understand so we can develop the therapies for. But you do need to look at the actual causes, and it's not just all in people's head. So yes, psychological aspects impact on whether a feature generates pain, but there's still a feature there that causes the pain. And that's my key point why we need to move away from non-specific low back pain.
And your talk, obviously it's your field of work, the disc, but would you consider it a large kind of contribute to low back pain, or just one of many?
It's one of many. So obviously within anybody's career, you can't investigate every single possible cause. You need people who are looking at the muscular charges, the ligament effects, the bone effects, the disc effects. There is individual patients where the start of the pain will be very different in those individual patients. And that's why we need that much better diagnosis and stratification first, so that then as the individual researchers who are looking at their particular areas develop the new therapies, we then have options for those patients. So I've been working on the disc for 20 years. I don't say that all low back pain is caused by disc degeneration. Well, chronic low back pain is caused by disc degeneration. It's not. There's loads of different causes, but at the moment, we still don't tell people what's actually causing it.
So moving just slightly away from the disc itself, how much variability of the function of the end plate plays a role in disc degeneration? So just slightly away from the disc. Is it worthy of investigation in combination with investigations of the disc as well?
Yeah, so the end plate is really integral into how the disc behaves and functions. So if the end plate, calcifies, so for example, then you're not going to get the nutrient supply into the disc. So that's going to affect how those cells are behaving in the disc. You need to look at the end plate in terms of stratifying those patients, and also, potentially developing therapies where you can decalcify those end plates and cause nutrient supply to increase again. So, like I say, it's not one particular area that is isolated. We need to look at the whole area, and I really do think that we need to talk a lot more as well. So people from all the different disciplinary areas and that work on the different areas, we need to communicate and actually share our knowledge between, so that really we can actually find therapies for patients.
So this is just going through, and I'm trying to bring these together, but would you assess the proportion of pain caused by the disc in an individual patient? Or how would you assess the amount of pain is coming from the disc itself?
So at the moment, I think it's a very difficult way of doing it. So there is obviously I'm not looking at developing the diagnosis, there's other groups doing that, but there's some really exciting work looking at improved imaging, for example, where it's been shown that you can look at actual lactic acid levels, so the pH of the discs, and that's associated with painful discs. So if you've got a lower pH and there's more lactic acid there, that's basically causing more sensitization of the pain, but you do see that with increased degeneration. So I think with improved kind of image and modalities, movement function, so the work that you do, Alex, I think is really important as well in terms of how, those discs and the muscles and everything move around. It all needs putting together so we can actually treat people better. I'm just trying to look at so many questions.
yes. It's fantastic how people are being engaged. This is great.
Do you want me to take some of the ones on the gel system?
Go on.
So the gel system at the moment, there's a few questions about, are we going to move this to human trials and what's the patient benefits and things. So at the moment, we are at the lab stage. We're trying to get the funding currently to enable us to do the animal testing, which is a prerequisite before we can get the regulatory approval to move into clinical trials. But all the results we've got so far in the lab are really, really promising, and it's really frustrating actually when you get not back from grants for no real reason. But so if anyone's got a pot of money and they want to fund us to do some animal testing, that would be great, because I really do think that this would be good for patients. I'd certainly have it myself. Not that I can be on the clinical trial because that wouldn't be ethical, but there's a couple of questions about the loading regime, but I think I covered that later on than the question came up. So it's looking at it's a sedentary lifestyle. So there's only kind of 4 hours of dynamic loading during the day, but then more static loading as we go through. So pretty much what I'm doing at the moment, we're sitting on Zoom all day and things. I don't know. Can you spot any other ones, Alex?
There's one I was just about to send through. Congratulating on the talk once again, but I was wondering, how long do you think it will be before stem cell and injectable treatments are available for patients with disc disease?
So there's a number of clinical trials going on at the moment. So there's the Mesoblast trial that's been going on in America, and I know they've just got regulatory approval to do trials in Europe as well. So that's where they're injecting stem cells into the disc and then looking at the treatment. They do seem to have some good early results, which suggest that they have good effects on sort of pain, etc. But there's not really much evidence on whether they're able to induce regeneration. One of my concerns of the direct injection of stem cells into the disc is, do they actually survive? Because the lab work that's been done prior to those studies haven't really looked at those cells in an environment that mimics the more degenerate disc. So that's some work that we're trying to do with our system in terms of actually culturing the cells in the gels with, low pH, low nutrient, low oxygen, increased catabolic factors and everything. And I think you need to look at those factors all together. So really interesting to see how those trials go on. But I don't know how long-term the effects will be. The other trouble with the trials at the moment is they haven't got good placebo controls. So it's also the assessment of those trials in terms of, are the effects they're seeing just the placebo effect, or are they truly from the stem cell injection? So I think it's really important when we do get to clinical trials, we actually have proper placebo controls for the surgery as well, so we can actually assess it, really, really well.
So this has been used in humans already in their care?
So not the system that we're looking at, but basically just the naked cells, if you like. So naked stem cell therapies is being looked at in a number of clinical trials at the moment, yes.
And do you know anything about the patient-reported outcome measures for this? I mean, does this treatment improve those outcomes?
So as I say, the work that we've been doing isn't at those stages yet, but the clinical trials that are ongoing, they haven't reported the results back yet. Apart from the very So I think the Mesoblast trial have reported the early stage responses, so sort of 3 months. They have shown decreases in osmostory disability and pain sensation, but as I say, it's not well controlled in terms of with the placebo controls. So whether that's purely a placebo effect or not, really they need to look at the longer term effects, I think, to see that.
So a lot of this, we look at the disc individual levels. Have we tried this on multiple levels? And was there a change in the outcome?
So I think all the clinical trials that are going on at the moment are purely single level at the moment. It's all of this therapies are very early stage. So normally with clinical trials, you always start on the simpler systems. They're very, very tight controlled on which patients they're utilizing as well. And they can only be on single levels. But I do think that it would have the potential for double level treatments in the future. The other area that you could potentially use, certainly our system for, where you're getting that improved mechanical support as well, is in individuals who are undergoing, disc herniation for one level, and they're showing degenerate changes in the adjacent discs. So you could envisage that this therapy could be applied to those adjacent discs to actually sort of halt any further degeneration and therefore subsequent prolapses. There's a question in the chat that I just want to take because it's something that we're just starting to look at about the role of low-grade infections. So there's quite a bit of controversy, I would say, around the role of bacterial infections within the disc. So there are a number of studies which have identified P. acne within the disc, but there's also a lot of groups which are saying that it's not there. We've just got funding from, it's a large sort of international training network called Disc for All. And one of the projects we're looking at is actually doing really good screening across lots of patient disc samples to see whether there are bacteria within the disc in discs which are collected in an extremely controlled manner so that we don't get contamination from the skin or things like that, and then looking at whether that infection could be the trigger for the catabolic responses that we see. So ask me in a...
I'm sure you can still hear me, but for me, you've just paused. And you're back.
Sorry, I don't know why my internet is dropping out today. Where was I up to, Alex?
I think the last thing you said was, if you ask me.
And what was that related? Was that in the low-grade infections?
Yeah.
Had I talked about the Disc for All project?
Yes.
I think we've probably got 1 more minute for questions because we need to stick to time. And can I just perhaps wrap up here and say thanks very much, Christine. It's been a fabulous talk and plenty of interaction. Maybe we've not answered every question in the chat, but if you've any other questions, I would suggest you contact Christine at Sheffield Hallam University. So I'd just like to have a big round of applause for the fantastic work that Christine's doing. And thank you all for attending the session and enjoy the rest of the afternoon. Thank you very much.